Hi everyone, my name is Pavitra V, Department of Chemistry. Today we are going to discuss a topic called nucleic acids. Basically, nucleic acids contain genetic informations and they determine protein synthesis. It is of two types, DNAs and RNAs. DNA is nothing but deoxyribonucleic acids, which is a master copy for most cell informations, and they will carry the genetic informations, and they are the templates for RNA. RNA is ribonucleic acids which transfer information from DNA and which is a template for protein synthesis. Nucleic acids situated in the nucleus in the form of genes, that is chromosomes. One gene can give you a synthesis of one enzyme or a protein, and enzymes determine external and internal characteristics of a cell. Nucleotides are the basic units of nucleic acids, which is formed by three parts. One is phosphate unit, and another one is sugar unit, and another one is heterocyclic amine base. So these are the three parts of a nucleotide. Once again, nucleotide is made up of phosphate, sugar, and a heterocyclic basic unit. So this is the basic structure of nucleotide, and the base and the sugar is formed by beta N glycosidic linkages, so like this NH bonds. The major difference between RNA and DNA is differ from its sugar unit used. So in RNA we use ribose sugar, that is C5H10O5, whereas in DNA we use deoxyribose, that is C5H10 and O4. So here we are using one oxygen molecule less, one oxygen atom less. So this is the structure, this is the difference at carbon. So this is the second position, carbon difference. Here OH group is present and here only H group is present. Nitrogenous bases are of two types, purines and pyrimidines. Purines is made up of 6-membered ring and a 5-membered ring, which is a double ring, whereas pyrimidine is a single ring made up of 6-membered ring. So these are the two types of nitrogenous bases. This is very important. Nitrogenous bases, further purines are divided into adenine and guanine, and pyrimidines are divided into thymine, uracil and cytosine. The major difference is thymine is present in DNA and uracil is present in RNA. The structure is very important. Adenine is denoted by capital A, guanine is denoted by capital G, thymine is denoted by capital T, uracil is denoted by capital U, and cytosine is denoted by capital C. So these are the nitrogenous bases. When we come to the DNA secondary and tertiary structures, sugar phosphate backbones causes each DNA chain to coil around the outside of the attached bases like a spiral staircase. So the base pairing, hydrogen bonding, occurs between purines and the pyrimidines. This causes two DNA strands to bond together. So adenine thymine will form a one bond and guanine cytosine will always bonded. Always pairing takes place between adenine thymine and guanine and cytosine, results in double helix structure. This is the double helical structure of DNA. DNA consists of two strips made up of sugar and phosphate twisted around each other and connected by nitrogenous bases. So this is adenine and this is thymine. AT will form a bond and GC or CG will form cytosine, and guanine will form a bond, and this will be a sugar phosphate backbone. So this looks like a spiral staircase or a twisted ladder shape. In 1953, James Watson and Francis Crick and their scale model for DNA, and they have formed a DNA structure. It is also called Crick-Watson model of DNA structure. This is the Crick-Watson model for a DNA structure which is having a base pairs and sugar phosphate backbone. So this is the major groove and this is minor groove. They will form two types of grooves in the structure. One is major groove and another one is minor groove. So this is one helical turn. So one turn will give us 10.5 BP per turn or 35.7 angstroms of length. So this is the helix diameter. The diameter is 20 angstrom, and this is the axial rise, that is 3.4 angstrom. So this is the structure of DNA. This is the structure of RNA. Here we have uracil instead of thymine. So here we will form a bond which is a single stripe, which is a helix of sugar phosphate unit, and this is the helix, whereas it consists of only one strand. This is the difference between DNA structure and RNA structure. This is the RNA, which have a cytosine, guanine, adenine and uracil, whereas DNA structure consists of cytosine, guanine, adenine and thymine. So it have a double helical structure, deoxyribonucleic acid, and which is having a single strand in RNA, that is ribonucleic acid. DNA and RNA have a major role in heredity, carrying from one generation to another generation. RNA and DNA are involved in three major processes in a cell related to heredity, as shown below. First one is replication, that is DNA copies itself. So one DNA will be giving another DNA by the process of mitosis. And the second one is transcription. The genetic code in DNA is written into the RNA and carried to the ribosomes by messenger RNA, that is mRNA. Next, the third process is translation, that is tRNA, transfer RNA, carries amino acids to the ribosomes as a part of protein synthesis. The protein will be synthesized by using a RNA transcriptions. So here transcriptions and translations are two steps in the biosynthesis of a protein, which is responsible for the growth of an individual. Chromosomes, these are the genetic materials carried out in the pairs. For example, the normal number of chromosomes pairs varies among the species. One species having a different amount of different pairs of chromosomes when compared to the other species. For example, human beings or Homo sapiens have 23 pairs of chromosomes, that is totally 46 chromosomes, 23 pairs. So cats having 30 pairs of chromosomes, mouse have 20 pairs of chromosomes. So these are some of the examples for the pairs of chromosomes present in different species.